Okay, good morning, everyone. We are going to wait 5 more minutes if anyone will join, and then I'm going to continue and talk a little about computational biology on CARC systems. Okay, good morning again. I think maybe we can start right now. So today I'm going to talk about computational biology on CARC with the use of OnDemand platform that we deployed 1 year ago. So let's get going. As a part of initiative to offer more resources for biology, last year we started an initiative called Bioresources. We are working to change that name. What we do offer in Bioresources is a set of reference genomes and databases. Those are updated twice a year. We do have genomes, mostly from Illumina project, a collection of BLAST databases from GeneBank, Genome Taxonomy Database, PFAM database, TIGRFAM, and UniProt. These mentioned are available from our website. You can get through that really easily. If you need particular BLAST database, you just choose the format and which database you need, and then you can copy the path and use it in program. However, we also have several databases that are not available through web interface. So you need to navigate to /project/biodb and check what is available there. So out of those, we have BioGeotraces. It is component of International Geotraces program. This is set of metagenomes. We also have Tara Oceans, set of marine microbial metagenomes. And if you are using VEP, Variant Effect Predictor software, we host Variant Effect Predictor cache that might speed up your computations. If you need any particular resources, please do not hesitate to get in contact so we can host it for benefit of everyone. So to get going, we can use terminal, but before this, on June 15th, we introduced the requirement that we need to connect to VPN. Personally, I'm using connectusc.edu, so I was connected right now, so I'm going to reconnect. With VPN, we can avoid constant duo questions with every login and increase the security of the connection between your computer, your laptop, and CARC cluster. That also allows us to offer a more broad portfolio of services. Some we are developing and should be available by the end of the year, like hybrid cloud platform. So once I logged into VPN, we can go to our CARC OnDemand dashboard. and try if shell access works from there. So I'm going to go there right now. We can go to user information, CARC OnDemand, and just click on login to get there. So once we are there, we can have direct access to Discovery shell and Davor shell. So it is that easy, even if you do not use a regular terminal application from your workstation. So to get going, I'm going to paste in a chat link with a small GitHub repository with the resources that I will be using during this short workshop. And inside of this repository, you will find the presentation I'm using, as well as the examples. So once we have, you can try to do the examples with me or just observe and then later try on your own. So once I got the examples cloned, let's get through the file management. There is plethora of ways you can manage the files. You can use WinSCP, CyberDuck, Pure Shell, or we come through web interface. If we choose web interface, we have two rows of buttons, like the more general one, where we can do some sort of navigation, and detailed one that we can utilize to manipulate with files. So if we click, it shows us what projects we have access to. Mostly it will be your home directory, Scratch directories, and one of the projects you belong to. So when I open the shell, by default, I was in my home directory. So let's go there and see if we have workshop bioresources copied. So it takes a moment to display the content. And yes, we do have something. There is a directory data with example data, jobs with sample jobs, and presentation that I'm using. So to follow through, the first example I'm going to go through is to do a simple NCBI blast. Of course, you can get things more complicated as you go. To do that, we are going to use jobs and job composer. Job composer remembers what jobs you are running. You can easily use it to manage your jobs. As you can see, there is something I was running before. So to create a new job, we need to use new job and choose the one from specified path. Because I was doing this before, it remembers the path I had before, but you have to find it out. the easiest way would be to just go to the directory and copy it. If you want to use a script from inside of directory, this / at the end is important. Name of the job, it is up to you. Name of the script, we have to see what are the examples I'm going to show today. So there is blast1.job. So this is something that I'm going to use. We are going to run job on discovery cluster, and as an important thing, you need to specify the account you belong to. If your PI has an account on our system, that is fantastic. You can use that account. If not, you can ask them to create one. Students are not allowed to have projects and accounts on their own. They have to belong to a group. So if you are a member of a group, you can type the ID command. That will show you all the groups you belong to. Among those you will see the accounts you are part of. Of course, you can type my account command. That will also display the list of accounts. I'm using this sysadmin and research facilitation special group called HPC root, but you have to put one on your own. So I created a sample job with sample script. It is not submitted. All I need to do is edit the submission script and check what is the version of SwissProt database. So in order to do that, I need to go to bioresources, check in GenBank SwissProt database. I'm choosing SwissProt just because it is small and the result will come in really, really fast. Of course, I need to adjust a few more things. And what is important is that this is the input data. Our query, it is a simple script. I'm going to show it. Data, blast, and query txt. This is kind of slow, so I'm going to use the terminal. So it is just short sequence without short protein sequence without any special formatting. However, the output goes to results blast, and that's the directory we don't have. So the base directory is set right there. And let's see how submitting the file will work. So you create a job, not exactly fully because the output directory is not there. What happens is your job starts. You can check that with SQ command and then type your username. The job is running. We can see the node. In fact, what the job is doing, it is waiting 20 seconds just so I can show that it is running. And then the job takes about 10 seconds to actually complete. The job is completed. However, when I check the output, there is nothing there. And output went to nowhere. That's why what has to be done is you need to make sure to create output directory. This time I'm going to remove this 20-second wait. And of course, I'm going to create a new one from selected job. I need to wait a moment for the submission script to appear. Submission script is there, already edited. So if I submit this job, it will create the output directory and then it will put things there. So if I check the job, is it running? Oh, it's already completed. As I can see, this one ends with 73. The output looks like this. However, if I check, we have a results directory we didn't have before. And inside of BLAST, we should have results file. So I can check that, oh, that sequence might have come from a mouse or maybe from human. They both contain identical protein sequence pattern. So that was the BLAST example. I have put this information inside along with the instruction how you can utilize the terminal and OnDemand to do so. So the next example is related to the command fastq. Fastq is used to do some quality checks on sequencing job. So I'm going to use the first job. It's fastq1.job. So again, create job from specified path. Script name is fastq1.job cluster discovery HPC root. Then I need to edit the submission script. Again, account name. Again, this script is waiting 20 seconds. Just to remember, the output directory has to be created. I'm going to do it right now. So, because this fastq example is really simple, I'm requesting just one node, just one processor. I'm choosing debug partition because that partition usually is not utilized. And since my job is really fast, that is the best partition to choose. And I'm requesting 5 minutes. So to actually check which partition currently is utilized, I'm usually going with sinfo2. So I can see that debug partition, even though it doesn't have many nodes, all the nodes are idling. The compute time limit is just 1 hour. If I went with main partition, as you can see, all the nodes are either allocated or in mixed use or some other state. It's completing job and PL&D. That's something that I do not see often. So you might wait for your job to run. The same goes for GPU partition. It is heavily utilized. So for short jobs, debug is the best choice. So having edited the file, created output directory, and provided the input, I'm going to submit the job. So I save the file, ready to submit, and it should take a moment to run. Again, I can check using SQ-U and my username. So the job is running, because it is waiting 20 seconds, so we can see how it goes. So one of the outputs of FastQ program is HTML file that I'm going to show. That is the biggest advantage of the job completed. That is the biggest advantage of OnDemand, because we can easily inspect HTML files. You can download the file. And get to see what quality was the input. And of course, the situation where you have just one file might not come very often. Sometimes you may have several other situations. So for that, I have other examples. Let's check out this one. So once again, I want to create a job from specified path. Name path to script name. And so in this situation, we have 6 files to analyze quality. For all intents and purposes, it is much more reasonable to create one script that goes through all of your samples than to submit 6 scripts, just because it is much better for Slurm to schedule your job at once, so all your requests will be handled at the same time, and you will be waiting only once in a queue and not 6 times. I guess I can remove the sleep thing, so I can speed this up. I'm going to create output directory, partition debug, and 6 runs. Even though I need to wait for the file to update. Okay, the file updated, so I can submit. Of course, this way it will run FastQ once, then second time, third time, fourth, fifth, and sixth. And your total wait time will be much, much longer. When I check the usage, it is just one job, and it takes 6 times the time that would be actually necessary. On the cluster, we have many nodes. Each node has many CPUs, so why not to take advantage of that? Let's check the output. So as we can see, 6 outputs, and utilize parallelism from the cluster. For that, we have arrays. So I have 2 arrays example. First is numbered array. So to do that, I'm going to choose the source path again, past q ordered numbering. What was the name? Script name, master discovery, count. Let's see. So if we choose to utilize array for our job submission, the submission script will change a little. Of course, it will still require, I'm going to choose debug partition because as we saw earlier, main partition was full. I know that I have 6 files, so I'm choosing array numbered from 1 to 6. Will then allow Slurm to replace certain ID in the naming with array ID. Going to put account name. Then I'm going to create the output. And actually, for this one, I'm going to leave sleeping for 20 seconds just to get a chance to show you how it will look when it's running. So the name of the files, I'm going to use something called slurm array task ID to substitute certain value from a file. So if I check how the file looks like, I think I'm going to use a terminal just so I can navigate slightly faster. So we have files numbered 1, 2, 3, 4, 5, 6. So it is totally easy to replace these numbers with array ID. If you have such a case, then your submission script might look like this. You put just one command, and you let Slurm take care of substituting the ID. I just save the file. Let's wait a moment for the interface to update it. And now I'm submitting. Job submit limit. Yes, we have certain limits on debug partition. I might have not realized that. So I'm going to submit to main partition and hope the job will run quickly. The job was successfully queued. So if I will check I see the job is pending. If I check again, it is pending. Actually, if I add dash dash start, Clerm will tell when my job will start. It didn't tell anything just because my job started. So as we can see, we have the main job ID and then RI ID, and we can see that the job actually runs 6 times. Of course, each of those is waiting 20 seconds. So it was longer than in reality. So if I go to results, I can see that I got exactly the same result as I got in the first time. And of course, oh, thank you, Luigi. Can I make it bigger? Oh, I didn't know. Hopefully that is. I'm going to change the terminal. This one is much easier to make it bigger. So for the last job, I'm going to use different terminal. And I'm going to go with unnumbered array. I'm going to copy the name. With name cluster discovery and HPC root. So this time, when I go to input data and check what names of files I have, it is anything but numbered. So there has to be a way to make this list a numbered list so we can utilize that with array IDs that, of course, are sequential. So what we can do is we can create a file with a list of these files. So first row will read name, second row will read a name, third row will read a name, and then we can utilize array IDs with row IDs and then read a particular row to do the substitution. In order to do that, we do similar thing. We request similar things. We request an array, and then, of course, we have to put our account name. This time, I'm going to remove the sleep. I have to check where the output goes. I have to make this directory. -p tells make dir command to create all the directories needed in order to create the last one. Sometimes it is very, very useful. So the example goes like this. We are setting the base directory to this. Then we are making, we can use a relative path number, a path to list the directory, that is the directory, and create a list of files. Then to prepare the input, we are creating for each array, we are creating a variable called line in which we are doing the substitution I told about. So we are reading specific line equal to slurm array task ID from the file we created a moment before. Then we are running fastq command with that file name that we read from particular line. So how does this work in a practice? Very simple. I'm going to submit the job. And let's see. The job is waiting. Usually, start time shows up after a moment. Or in this case, the job just started. And it's completed. CG states for completing state was still running, so they all pretty much ended at the same time. So if I go to results, they look exactly the same as other results. And the submission script worked with that pretty well. Of course, not every example is that simple. So the bigger one I have, the bigger exercise is to go with genome mapping, to actually read the mapping and show that on a genome. So how do we do that? We have to use several tools. We can use FastQC, Bowtie, Bowtie 2. Each of them is best at its own purpose. So it's not like Bowtie is outdated. Bowtie is best for short reads. Bowtie 2 is better for longer reads. We have BWA, then we have SAMTools package that we can utilize to convert binary file into non-binary file and back. We can extract particular elements like certain fragments to a different part. Why? Because genome files are huge. Processing huge files requires lots of resources. And of course, it requires a lot of time. So if you run things once, then it doesn't matter. If you run things several times, then time savings are sort of critical. So for example, when you do the conversion of some, you can use switch lowercase b to specify that you need binary file, capital S, that you have to tell SAMTools that you are reading some file. If you utilize -h, sometimes for programs it shows help. In this case, it shows to create proper header. Sample readout output is like this. It tells how many duplicates happened, what was the quality. Then you can utilize several other tools from samples package to create FASTA index just to speed up reading large FASTA files, or merge several BAM files into one. Of course, we have other tools to manipulate genomes. We have bed tools. So we can create several views of a genome. In examples included in this presentation, you can see how to create an overlap or non-overlapping section, or what reports the intervals that represent overlaps between two files. So in the exercise, we have some 2 paired reads of DNA sequencing. Those are very small ones, so it will take not too much time. I have a mistake here, so please disregard this and go to data to check on these 2 files. And we'll study particular region of human genome. Then we will read we'll map those reads to HG19 version of human genome. Of course, we are going to extract only part that map to our region, and then we can utilize UCSC Genome Browser or Interactive Genomics Browser that we offer as one of our interactive apps. And check what happened. Of course, we are going to count number of insertions and deletions in a SAM file. So how do we do that? So we need to load bowtie program. After this short workshop finishes, you can go slowly through the presentation and check why we are doing certain things. So when we load bowtie program to play with the reads, we are going to copy sequence of chromosome of our region that interests us. That's chromosome 21st. Then we are going to map our reads with bowtie 2. Of course, we need to build an index. Then we need to do number of conversions, so we can count number of insertions and deletions. After that, we need to extract the reads with bed tools, create tab delimited file with coordinates. All the tasks that are necessary are listed here. Of course, they are not. I'm not providing commands here. That is for you to figure out. However, I'm providing also the example script, so you can always start by analyzing the script. So let's go with that. Again, creating a job from specified path, name the script name, cluster discovery, and HPC root. So in a moment, the script reads, and I'm going to edit that. So loading a bowtie, that shouldn't be complicated, of course. I'm going to adjust the script first. and put account name, my username. I'm ensuring no other modules are loaded, loading one of the compilers we have, and then loading Bowtie2, SumTools, and BedTools2, as those are the tools I'm going to use. So first, I need to create the output. So always check for that. Then I'm going to copy the sample files. So those are in data, R1 and R2. I'm going to unpack them. Now I have to check the chromosome if that is the correct path. Maybe it changed. So choosing name, Homo sapiens. So it's HG19. Chromosomes, chromosome 21st. We have a path, so I'm going to click and copy that and ensure it is correct. Yes, it was correct. Now going to create a Bowtie2 index. The number of threads to utilize more things that I'm going to use is I'm substituting it with Slurm and tasks. I have had 20, but because main partition is sort of occupied, I'm going to put 5. You always want to use variable substitution, just so if you change the parameter in your request, it gets copied in every place you use that. So creating an index, providing the input in fact. In fact, I can use this chromosome directly. I don't have to copy that. And instead of copying, it might be much better to create a symbolic link. This way, it will still look like you have the chromosome, the file of interest inside the directory you want, but you will not use your quota. So after building the index, I can do the conversion to SUM format. Utilizing this index. So example first, example second. Then I can count number of insertions and deletions. And the field number is 6. So this is how the command looks like. So after that, going to the second part, we are utilizing BedTools2, and we need to create tab delimited bed file. So in order to do that, we have to convert SUM to BAM. We need to sort the BAM files, create an index, and do the extraction of the region of interest. And now to extract the reads, I'm going to use bad tools intersect. And that concludes this example. So how it works in a practice. I'm going to submit the job. And see how that goes. So I was lucky, my job started immediately. So I'm going to check on the reason. On job ID, it is 118847. So I can 17. Which one is that? That's the output file. The job is still running. So if I go to results, I can see that the last file that I wanted it to produce, called reads bad, is already produced. So the job, yes, the job finished. Just as I said, when I created symbolic link, it looks like a file, but it points to the original place. So how do I read this file? Of course, I can submit that to UCSC Genome browser, but I'm going to use interactive genomics viewer to map that region, to read that mapping. So I'm starting interactive application. Of course, you need to provide an account, partition. Because it is simple, I'm going to use the default values, and I do not need any constraints. So let's see how that goes and how long I will have to wait to start my application. It is still waiting. I'm going to check. Okay, so my session is running already. So I should get the link immediately. Slightly impatient. So I will refresh that page. Yes, and the link is there. So this is standard application. There is a bunch of genomes you can load. We can load them from file, and to load them, we need to go to project. Then we need to scroll a little to find BioDB and go to IGV and genomes. Because I loaded the genome before, it is available, so it loaded automatically. Now I can load a file. If I go to read mapping, there is a reads.bed. When the file is loaded, should be able to double click on it. Chromosome 21st. And we can see that it's loaded. So how do we do that if we want to utilize if we want to utilize UCSC Genome browser, the one accessible from the web. So we need to download reads.bed file locally. So we need to go to our results. I do not know why the interface is slow, but sometimes it happens. I will need to get in touch with our web admin. By resources. Results. Read mapping and reads.bed file is easy. Okay. I might have been clicking, double clicking. Being impatient does not pay off. Okay, so I can save the file locally and then just go to UCSC Genome browser. We can click Add Custom Tracks. Going to choose H90 Genome. Load the file. Submit, and it already told me that it's chromosome 21st. Click on Go and see our region. It is quite similar to what we can see in Interactive Genomics Viewer. So that concludes this workshop. You are more than welcome to try it on your own, to check different parameters, try to break it. Of course, there are additional resources. You can read more about Slurm in those links. Please visit our forum. If you have questions, you get answers, and then it will stay there for others to help. Submit a ticket if you need anything. And please, if you have any questions, get in touch. So that's mapping exercise. And that is it. Thank you, guys. Do you have any questions?
Tomek, do you put this one in the YouTube or upload your PowerPoint or something? I missed something.
Yes, I'm recording it. So I'm going to ask our technical writer to get the link from that.
All right, thank you. Would you send the email? Then let me repeat again.
No, okay. So I'm going to make a list of people who are here, and then I'm going to send the link in email.
Okay, thank you.
Okay. Thank you for being here.
Thank you. Goodbye.
Bye.